Hello everyone, my name is Arthur. I am a biology tutor here at Art Education. Today I am going to quickly go through a biology topic in IBDP high level syllabus with you, and afterwards we will spend some time looking at an exam question related to the topic. So time is running out, let's get started. Today, the topic I picked is the linkages, as you can see on the screen. Okay, so based on what you learned from GCSE or IB and YB, you should know that alleles are inherited independently. This theory was proposed by Mendel, who was an Australian monk, and he used pea plants to demonstrate his finding. So first of all, we will have a quick review of what he did. I'll get to refresh our memory. So he used two pea plants that are heterozygous for two genes. The shape of the seed and the color of the cotyledons. So when we do crosses, we please remember to use capital letter for the dominant allele and small letter for the recessive allele. So here, the dominant allele for the seed shape is round, so we use the letter R, while the small r would represent the recessive allele, which is wrinkle seed shape. For the color of the cotyledons, we use yellow. Okay, the yellow one is the dominant one, so we use capital Y, and the recessive allele is green, so we use small letter y to represent. As both of the parents used are heterozygous, are both heterozygous, so the genotypes of the parent would be capital R, small r, capital Y, small y. Okay, so because we know that alleles are inherited independently of each other, both parents should be able to produce all sort of gametes. Okay, all different combination of gametes. We have capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now we have figured out the possible gametes. We can do a cross together. First of all, we need to see how many different types of, how many phenotypes there are. I can see that round seed yellow cotyledons is the first one, and the second one we can see that round seed green cotyledons, and we can see wrinkle seed yellow cotyledons, and finally we can see wrinkle seed green cotyledons. So these are the four genotypes that can be produced from this cross. Now, second thing we need to do is that we need to count how many there are. For the round seed yellow cotyledons, there are 1, 2, 3, 4, 5, 6, 7, 8, and 9. While the round seed green cotyledons, there are 1, 2, just 3. For the wrinkle seed yellow cotyledons, there are 1, 2, 3 also. Okay, so we can come up with a ratio of 9 to 3 to 3 to 1. And we call it the Mendelian ratio, as this is backed up by the Mendel's law of independent assortment theory. However, not all dihybrid crosses result in this ratio. The reason of this is because of gene linkage. So there are two types of gene linkage. The first one is sex linkage, and the second one is autosomal linkage. So sex linkage refers to genes located on the sex chromosomes. In humans, it is X and Y. But it also does autosomal linkage refer to genes located on the same chromosome other than the sex chromosome. An example can be shown below. So the enzyme, the gene that encode for enzyme trypsin and the gene that encode for CFTR protein are both located on chromosome in humans. In this case, we say that these two genes are linked, and so they do not, so they're not inherited independent of each other, as they are both located on the same chromosome. And now an example can be shown here using sweet peas. Here, there are two different genes that encode for its flower color and pollen grain shape. Here, we would use capital letter P as the dominant allele, give rise to a purple flower color. The small p represent the recessive allele, which is the white flower color. And for the shape, we have capital letter L for the dominant allele and small l for the recessive allele, which will give rise to short pollen grain. Now, the genotype here is, we have to be careful with the notation. Before, in the pea plant cross, we use, we just wrote the letters out. But for gene length crosses, we have to either use a line to show that these alleles are connected to each other. Or you could use brackets to show that they're linked. So you would have to use notation like this. So if the genes are unlinked, there would be 4 different possible gametes produced. So as you can see here, but for linked genes, there are only 2 possible gametes that produce. 1, 2. And it's same for both parents. This will make the cross a lot simpler than the cross we did earlier. And there are only 2 phenotypes produced from this cross, which is purple, long, and white shorts. The ratio is 3:1, which does not comply, which is not the same as Mendelian ratio. But in reality, there are actually some small numbers of purple short and white long offspring produced. The reason why is because of genetic variation, genetic recombination, sorry. I'm not sure if you can remember, that's crossing over occurred in prophase 1 between homologous chromosomes. When in prophase 1, genetic materials, some of the genetic materials, so we have non-sister chromatids crossing over each other, making a chiasma. And when they are pulled apart, there will be exchange of genetic materials across chromosomes. So that's why new gametes can be formed in this way. So so if we have 2 dominant alleles on the same allele, on the same chromosome, and 2 recessive recessive alleles on the same chromosome, these can be exchanged to form capital L small p or small l capital P. This explains why we have these different variations in the offspring. So scientists also found out that the greater the distance between the genes, the more likely a crossover will occur between them. An example can be shown here. So in chromosome 7, there are a couple of genes in there located on there. Let's take PSP, EGFR, and CF gene for example. Okay, so the distance between PSP and CF gene is greater than the distance between EGFR and CF. That means crossing over is more likely to occur between PSP and TCRB than between EGFR and CF. Okay, so scientists can use this information and the frequency of recombination to map the genes on chromosomes. Okay, for example question, let's have a look at this one. So autosomal genes are located in the chromosomes that are not sex chromosomes. The inheritance of autosomal genes is affected by whether the genes are linked or unlinked. Explain 2 types of inheritance using examples of parents that are heterozygous for 2 genes, A and B. So here, you will have to explain the 2 principles of the principles of 2 different types of inheritance. Okay, it's strongly recommend that you do a Punnett square cross to illustrate your understanding. Okay, it's like what I've done here. So here you should so for unlinked genes, the parents should be able to produce all different combinations of gametes, and the ratio of the phenotype should be 9:3 to 3:1. Okay, you should also explain why. It's because unlinked genes are sorted independently. Okay, now after explaining the principle for the unlinked genes, you should talk about linked genes. So you can say that it does not is unlike unlinked is different to unlinked genes, that it will produce non-medallion ratio. Okay, there are only 2 types of gametes they can produce. Okay, and however, even though there are only 2 types of gametes produced, okay, crossing over could create genetic recombinants. Okay, which allows new gametes to be formed. You should also make a point that the further away 2 genes are on the chromosome, the more likely there's a crossover between them. Okay, this is pretty much it from me, and I hope you enjoy this video. If you find this video useful, you should arrange a trial class with me here at the Arch Education. Hope to see you soon all. Goodbye.